Hello everyone. We will continue with our last video on pharmacodynamics series. So in this video we will talk about combined effect of drugs. So first thing is why we want to study this combined effect of drugs. Because there are many instances where drugs are given in combination. Now this interaction between two drugs, they can be of different types. One, either the drugs will not interact at all, or there can be synergism, or there can be antagonism. Synergism is where the response of one drug is increased by the other drug. So that is synergism. Antagonism is where the response is decreased. Okay. So synergism can be of two types, additive or supraadditive, which is also known as positive synergism. So additive is when the effect of both the drugs combined is equal to when we see the effect of one drug and the other drug and both are combined. Say suppose we'll take some arbitrary numbers. Say A alone is having x1 effect and B alone is having x2 effect. And we add this, so what will be the total effect? It will be x1 plus x2. So when we give both the drugs together, then the effect which occurs is equal to this x1 plus x2. On the other hand, in supraadditive, what we see that when these two drugs are given together, effect is more. So this effect will be x3 and it will be more than x1 plus x2. Okay, so that is supraadditive. Then there is antagonism. In antagonism, what we see that the effect of these drugs is lesser. So x3 is there, which is lesser than x1 plus x2. That means one drug is opposing the action of the other drug. Okay. So these are the various types of interactions. Now, these type of interaction we can study from our dose response curve. So this dose response curve we have already dealt in our video on dose response curve in pharmacodynamics. You can have a look on that video as well. So here this graph is showing dose response curve where x-axis is showing log dose and the y-axis is showing the percentage of response. And say suppose this is drug A and this is the response of drug A. Now, this is drug B. Now we see that normally this drug B does not has any response. But when it is combined with drug A, then we get this response. So what is this? So what is happening that normally B is not having any effect, but when it is given with A, it is decreasing the required dose of A. Say suppose this is 50% of response, okay? And when A is given alone, this is the dose which is required. But when given with B, now the dose required is much lesser. Okay, so this is decrease in the dose of the drug A. Now this is known as synergism, because you see that to say suppose this is X and B response is 0 when it is given alone, then it is X plus 0. But what we see that this B plus A, now the response is much more and dose required is much lesser. So that means the final response is much more than when the drugs are given alone. Okay, so this is what? This is synergism. Now say suppose this is drug C and it is having its own dose response curve like this. But when we combine C and A together, there is a shift in the curve, but the response is additive. How to determine that? Say suppose we take this particular dose and with A drug there is say 20% response and with drug B say suppose there is 30% of the response. So when we combine C plus A in the same dose, the response will be this much. So 20 plus 30% of response will be there. So that is additive kind of interaction between the drugs. Now next kind of interaction is antagonism. That also we can see in dose response curve. In competitive antagonism, we see shifting of the curve to the right. Efficacy is not decreased. That means the maximum upper limit which is there, it is not decreased. However, the dose of the drug required, that is drug A, dose required is increased. So you see that initially with this particular dose, this much response was there. Now, only this much response is there. So that's why another drug has antagonized the response of A. However, if we increase the dose, we get the maximum response as well. On the other hand, in non-competitive antagonism, we see there is shift in the curve to the right and also the maximum response is decreased. That means even if we increase the dose, then maximum efficacy is not reached. Now finally, we will talk about something known as isobolograms. What is isobologram? Isobologram is a graphical method to study interaction between two drugs that have effect on same physiological phenomena. That means they are having similar pharmacological effect. So here in this graph we see on x-axis dose of A and on y-axis we see dose of B. Now what we do, that say suppose we plot at what dose these drugs will have 50% of the effect. So that is what? That is EC50. Okay, it's a concentration at which the drug is having 50% of the maximum effect. So say here when we are using A alone, this is the dose which produces maximum effect. So B will be 0, A is this dose. And then dose B, say this is the dose which produces the 50% of maximum effect. Now we draw a line which is connecting these two dots. This is known as line of additivity or isobol. Okay. Now what we do, we give these drugs in combination, that is A plus B, and see that which dose combination is going to produce 50% of the effect. Okay. For example, we give dose A 1 gram and dose B 10 grams. Say suppose it produces 50% of the effect. Then we change this combination. Now we give A 2 grams and B we give 8 grams. Then maybe here we give 3 grams and 6 grams. And all these combinations produce 50% of the effect. Now this combination we plot in this graph. Say A 1 gram and B 10 grams. Say it comes here. Then A 2 gram and B 8 grams, say it comes here. Okay, so there will be various plots. Now we join all these points together and create a graphical line. Now based on where this line is coming, we can determine how is the interaction between the drugs. If the interaction is additive, then all these points will come on this line itself. Okay, so say suppose drug B this much dose will be required, drug A this much dose will be required for 50% of the effect, drug B this much dose and drug A this much dose for 50% of the effect. So all these points if you join and the line is coming coinciding with this line of additivity or isobol, then the interaction between the two drugs is additive. If the line is coming below this green one, then it denotes synergism. That means the dose of A and B which is required has decreased than required in case of additivity. And antagonism, that means now the graph will come up and the dose of A, suppose dose of A is antagonized by dose B, so dose of A required is much more. Okay, or if both are antagonized, then both dose required is much more. So that is line of antagonism. So what is the point of studying this synergy and antagonism? That suppose if the drugs are antagonizing each other, that means the required pharmacological effect for therapeutic efficacy, what we require, that is going to decrease. So we have to be careful. We may need to increase the dose of certain drugs. And secondly, with synergy, it may be that the pharmacological effects which are required for treatment, that may add up, but side effects don't add up. So in that case, it is better to use two drugs together rather than increasing the dose of one drug to increase the therapeutic effect. Understanding, say suppose if we want analgesic effect and we are using one drug. So if we increase the dose of one drug, along with that the side effects are also going to increase. But if instead of increasing the dose, we combine it with another analgesic drug, then the effect is going to increase and need not increase the dose. So that was about combined interaction of the drugs, and with this we finish our pharmacodynamics series. Thanks for watching the video. If you liked it, do press the like button, do share the video with others, and don't forget to subscribe to the channel Physiology Open.